Right, good afternoon, everyone. My name's Simon Richardson, and I'm here to talk to you about some of the things my lab gets up to. So to advance the slides, I do that. Oh, splendid. Quick disclosure statement, just to keep me up the straight and narrow. I'm going to talk about some work which is covered in two patents, which you can see here. One is involved in the use of attenuated anthrax toxin to get large polar molecules into cells. And the other one is concerned with loading the same molecules into exosomes. Right, so compartmentalization is something that I spend a lot of time thinking about. And a great example of this that I tell my students when I'm teaching them is the fact that I'm able to sit here, or stand here as the case may be, and carry out mutually exclusive operations. One would be an anabolic and one would be catabolic. For example, the digestion of my rather lovely lunch in my tongue. I'm thinking about hopefully entertaining and educational things to tell you, my audience. What would happen if the contents of my stomach got into my cranial vault? Well, it'll be entertaining for you, and we'll probably end this talk fairly quickly. Yes, I have what little is left of my brain degraded and digested. Cells do exactly the same thing. So here we have a picture of some Vero cells. Oh, there they are again. And these cells have been fed wheat germ agglutinin, which has had Texas red conjugated to it, which is quite a lovely fluorophore. And then we fix the cells, and then stain them, immunostain them with an antibody called LAMP1. LAMP1 is lysosome-associated membrane protein. So here we have cells that have been fed wheat germ agglutinin, which has been chased into a late endocytic compartment, and we can verify that by staining those cells with LAMP1, which is a very well-known market-fed compartment. And this is the cell study. So what would happen if the contents of the cell study got into the nucleus? Same thing that would happen if the contents of my stomach got into my cranial vault. It wouldn't end well for the cell. Consequently, the cell has invested quite a lot of energy in the regulated movement of compartmentalized material. Compartmentalization, that's where I was going. Consequently, we can see in the cartoon, we're reminded from the micrograph that A and B are not the same. They're separated by a barrier. And that would be endomembrane, or the plasma membrane at the top. And this intracellular compartmentalization is kept in steady states by processes such as endocytosis. And this is required for membranes to provide a compartmental barrier to keep the contents of lysosomes within lysosomes and not chewing up the nucleus or nuclear material to turn the cell into soup. So a while ago, I was thinking, okay, this is kind of cool. How do cells move things across compartmental barriers, for example, during lysosome biogenesis? And are there any other materials that take advantage of these very highly regulated trafficking steps? And the first immediate answer is viruses. Well, we know an awful lot about the use of viruses for gene therapy and the delivery of nucleic acids. But if we just deconstruct viruses, we get back to things like protein toxins, which you can manipulate using molecular biology recombinant technology. And that's thought that this affords the opportunity to unscrew the warhead from these protein toxins and replace it with a nucleic acid or cargo binding domain that can be used to transport material across these membranes into the cell. So we tried with ricin toxin, and it was a disaster. It didn't work at all for lots of different reasons. And we tried sugar toxin, and we tried cholera toxin, and none of these were particularly successful. These take quite a circuitous trafficking route to the cytosol and are very, very potent toxins, so they don't need efficient trafficking. It wasn't until we hit upon anthrax toxin that we actually had a degree of success. Anthrax toxin is very, very interesting in that it uses an airlock to access the cytosol. And you can see this airlock here. This is called an intraluminal vesicle, and it's used for destroying activated mitogenic receptors. So we're hijacking the system with anthrax toxin, and we've taken the B chain, PA83, which is then hydrolyzed to PA63, which are oligomerizers, and then in an acidic environment, drops the receptor and inserts into the membrane. And instead of using lethal factor, we're using non-lethal factor, which is lethal factor that's had domains 2, 3, and 4, which are the catalytic domains, removed and replaced with, initially for siRNA, the RNA binding domain of human PKR. we were delighted to see that we got quite a lot of transfection when we initially tried this, and we can import an array, a mismatched array of antisense oligonucleotides and also siRNA using different derivatives of this LFN protein, which pulls these molecules through the pore into the lumen of the ILV, which will then backfuse with the limiting membrane of the multivesicular body, releasing cargo into the cytosol. So this is the general scheme of it. We can make these proteins recombinantly. That's not a problem at all. We first published it in about 2015 in the Journal of Controlled Release, which is the Dyer paper. We've recently published the application of these things targeting Zika virus in virus genes, which if you were to Google my name and Zika virus and anthrax toxin, I assure you won't get arrested, but you may just find the paper. Right, however, it's not quite as simple as that, because one of the reasons we were able to patent this system is because it operates in a non-obvious way. So here you have a top-down view of the anthrax toxin. This is a side view. This is the bit that's inserted into the ILV membrane. And this bit here is called the phenylalanine clamp. And it has a diameter of about 6 angstroms. What? Hold on, you just told us you've got siRNA through this thing into the cytosol. It's also a cation selective pore, if you believe the literature. So I think what we are starting to think is that possibly this isn't a static structure as the literature would have you believe, but in fact it's a breathing system. But can we actually test that? We've tested the PA that we make in a recombinant way using E. coli, and we've fired neutrons at it, as well as done a variety of other sort of analytical things. And we are reasonably confident that it's about the same size as the models on PDB, and it seems to be behaving in a predictable way, which makes us fairly confident that we are actually doing what we think we're doing. So can we deliver siRNA with minimal toxicity? Yes. We've seen in some cells that we can do this with an efficiency similar to nucleofaction. Hooray! Could we be bursting the endosomal membrane? Well, my background is using linear amphiphilic polymers to deliver stuff and burst endosomes. And that was during my PhD, which now seems to be in the mid-Jurassic period. However, a lot of that information is still stuck. And I think asking this question, are we instead not contradicting some of the established literature, but maybe we're doing something we're not intending, like bursting this endosome, which is a system that's happening irrespective of ILVs. Can we test that? What happens if we inhibit ILV formation? We've seen from the literature that if we can inhibit ILV formation and specifically back fusion, that we can prevent up to 80% of lethal factors activity in cells. We know that from the Vandergrift lab. So what will happen if we inhibit PA? That's asking another question about specificity. So Paul Luzio's lab up in Cambridge have on many occasions published the use of sucrasomes. This is taking late endocytic structures, loading them up with hypertonic sucrose, which increases the luminal osmotic pressure, preventing the formation of ILVs, as you can see here and here. We've also used wheat germ agglutinin in quite a lower concentration than this, and you can see the appearance of these large bloated endocytic structures as we fed these cells Texas red wheat germ agglutinin, and you can see that there's not much in the way of internal membrane there, saying that this wheat germ agglutinin, this cross-linked membrane, is stabilizing that membrane to the point where ILV invagination is really against entropy. So we've looked at both of these techniques and we've asked, can we not only get siRNA into the cytosol, but can we get something that's faster to measure? And in this instance, we've used lethal factor, which is the anthrax pore entry sequence, used in frame to the diphtheria toxin A chain, which inhibits elongation factor 2. So without any PA, LFN DTA is not exhibiting a huge amount of toxicity. However, the moment we put PA on there, voila, we get PA concentration dependent cell kill. This is in HeLa cells. So this is a measure of, are we actually getting stuff over the PA pore? And we can assay for siRNA activity using our system, or we can assay for cell viability using LFN DTA. We've also got, this is the assay that we've developed for siRNA activity. We're using stably transfected cells. which are HEC cells, which are stably expressing emerald GFP fused in frame with beta-galactosidase and then bisystrologically expressing RFP. Now that beta-galactosidase, we all know, will hydrolyze XGAL from a clear solution or a white solid to something that is bright blue, which we can measure spectrophotometrically. And that's what we've done. We've asked in these cells, if we get an siRNA response, and we've directed our siRNA against GFP, because that's in frame with beta-galactosidase, will that downregulate beta-galactosidase activity? We can measure it on a spec. We don't have to do RTQ, we don't have to do Western blots. It's quite rapid. And that's exactly what we did. So this is where we put diphtheria toxin A chain in the cells that have had concentration-dependent wheat germ agglutinin added to them. So this is the lethality we would expect normally, where our PA is allowing the DTA through the PA channel into the cytosol, killing lots of cells. As we increase the amount of wheat germ agglutinin we give the cells, we're seeing concentration-dependent inhibition of DTA lethality. Ooh, okay, cool. Similarly, we can see siRNA activity, which is also being reduced as we add more and more wheat germ agglutinin. Have we stopped endosomal limiting membrane being burst? Well, this is a cross-linking agent, and this probably doesn't address that, because it could be that the wheat germ agglutinin is really stabilizing the limiting membrane. So adding more and seeing less activity doesn't really answer the question. However, if we were to put hypertonic sucrose inside of these ILVs, we're increasing the intraosmotic pressure, and that's going to be a further stress on the membrane. So if we were seeing an endosomal lytic event, we would expect this additional stress to actually perpetuate that. And as you can see, we're not seeing that at all. We're seeing, as we add more sucrose, we're seeing less LFN-DTA mediated cell kill and less siRNA activity, which means, in all likelihood, this isn't just an endosomal lytic effect, but we are indeed inhibiting ILV formation, and we can see that microscopically and quantify it. So this isn't necessarily an endosomal lytic effect. It's, in all likelihood, an effect driven by the passage of these effector molecules, or drugs, if you like, through the PA pore. What happens if we inhibit the PA pore? Well, we recently discovered that there is an inhibitor molecule which is extremely thermodynamically stable, which I'm not sure I can actually name it at the moment, because we may be patenting it in the not too distant future. And this has LFN on it, and then very, very thermodynamically stable, big beta barrel. And that will enter into the PA pore. The literature says this does, it doesn't, does it? We don't really know. Does it go through the PA pore or not? We're not really sure. Anyway, when we put the cells in with this molecule and then added LFN-DTA, you can see there was a profound effect on cell kill, i.e. there wasn't any. Well, there was a little bit, about 20% wiggle room there, but relative to the no inhibitor, you can see that there is quite a profound difference. Similarly, if we put them all together with no temporal space between adding the inhibitor and the LFN-DTA, we again see quite a lot of inhibition relative to the non-treated control. Okay, so if we inhibit the PA pore by adding this very thermodynamically stable molecule that can't unfold, and that unfolding is thought to be needed to get through these phenylalanine clamp that I was talking about earlier on, we can effectively inhibit the passage of things that we do know get through PA pore. Okay, that's kind of cool. And we get direct evidence of our siRNA within the cytosol. And we decided to answer this question with a 32P label our siRNA using a 5' end labeling kit, being very careful to ethanol precipitate and also phenylchloroform extract our labeled siRNA to make sure that we could quantify how much free label was in there. And we were at less than 10%. We thought, oh, okay, we can live with that. So we put this 32P labeled siRNA and the PA and the LFN-PK on our cells, and then fractionates them. We have different doses, and you've got different subcellular compartments here. Here you've got the nuclei, then you've got membrane, then you've got cytosol, and we know from previous work that anything associated with the membrane and inside vesicles will be in the membrane compartment. So you can see here what is effectively cytosolic 32p siRNA. Don't be fooled by this large, the nuclear compartment. That's what I'm looking for, because this is due to the way that we've diluted hot RNA with cold RNA to get different doses, which would alter the way that you see these data. So I have to point that out, but I think here looking at the cytosolic fraction, that yeah, we can most definitely see siRNA in there. And that's a more or less direct measurement. Were that RNA to be chewed off, it would diffuse, were that RNA to be chewed up and the 32p released, it wouldn't remain in the cytosol, it would have diffused into the cell culture media. So how's this function next to different transfection methods? So we have a look at LFM PKR, PA, and oligofectamine 2000, and you can see the performance is, reasonably equivocal, which is good. We also look at knockdown using RTQ and knockdown using Western blotting to try to correlate the level of knockdown we see to the number of molecules per cell per compartment. And yeah, that data is there. We're in the process of writing this up at the moment for publication. So unlike many non-viral siRNA delivery systems, PA uses an ILV airlock to access the cytosol. That's important because it circumvents any possibility of membrane damage, which is something you often see with polycations like polyethyleneamine or polylysine, for example. However, it's possible some cells are more susceptible to anthrax intoxication than others, due to the fact that ILV biogenesis formation is very, very highly regulated, but all cells don't actually behave the same way. So we thought, wouldn't it be useful, especially if we're planning on using this as a therapeutic, to actually get a handle on this and to start that conversation? And that's what we did. So again, we took a panel of cells, we started by feeding them PA and LFM diphtheria toxin A chain. You can see at 24 hours there's a little bit of cell kill. However, if we increase that time to 72 hours, you can see that there's quite a profound difference between the toxicity profile in HeLa and the toxicity profile in A549 and U87 cells. Okay, we can rank that sensitivity more or less, but why might this be? What's responsible for it? Is it receptor expression at the cell surface, or is it actually the expression of proteins that may form part of the escape complex, which are actually responsible for ILV biogenesis? So the first thing we did was a very, very crude experiment just to measure doubling time. Is it having more effect on HeLas because they're much more membrane out, they're much more metabolically active than the other cells? They're growing faster, with the higher propensity for things like membrane misfusion effects. So we looked at that, and I think at the moment it's very difficult to pull a definitive answer out of this because we need to repeat the experiment a couple more times to shrink down our error bars a bit and to allow us to do meaningful stats. So we started to look at does this jump out? HeLas 24, these guys 48. These are the guys that were recalcitrant to intoxication. It's very tempting to speculate, but until I have more data, I wouldn't dare jump into that particular pool of parameters and things. What about the expression of receptors? So here we have the receptor 10-8 and the receptor CMG2, which are known collectively as anthrax receptor 1 and 2. And we're looking at different expression levels across cell length. Surprisingly, A549s, which don't really respond very well to PA and LFM DTA, have quite a high level of expression. And yeah, the receptors don't really seem to be key in all of this. However, if I was a betting man, seeing A549s, which are the least receptive or susceptible to PA and DTA, LFM DTA, cell death, having a reduced level of TSG101, which is a key protein within the escape complex needed for ILV biogenesis, it's tempting to say, well, I think this deserves a little bit more attention in the future. So I'm going to finish at this point and come to some conclusions. First of all, it's possible to directly quantify cytosolic 32p siRNA in a variety of cells after transfection of PA, LFM, PKR. These data indicate that the PA, LFM, PKR can import siRNA into the cytosol. What I haven't shown you is that it's also possible using a native page gel to recover that siRNA afterwards with the 32P label on it. It looks really funny, which is why I haven't shown you it, but nevertheless, the 32P is still macromolecular, which is good. The use of wheat germ glutamine and hypertonic sucrose as ILD inhibitors indicated that endosomal lysis is not occurring, which is further evidence that says, yes, our macromolecules that the literature says shouldn't go through the PA port are, which is great. And also, endosomal lysis is not occurring, so this is actually a PA port dependent effect. The ability of the PA port to move macromolecular cargo is further validated by the observation that there was variation across multiple cell types, again, lending its notion towards type regulation. RT-qPCR has indicated that TS2-101 from the SP complex is probably involved in this differential sensitivity. However, we need to gather more evidence before we can really hang out on that conclusion. It may also mean that some cells and conditions are likely to be better candidates for treatment with this potential therapeutic than others, which is why I think it's important to know this fairly fundamental information to start with. Next question on my mind is there anything else at play? We've seen plenty of evidence of it, oligonucleotides can, in a relatively inefficient way, get into the cytosol without any help. What happens if we put LFN-DTA on its own or ricin toxin A chain on parallel cells and see if there's a difference in the level of cell kill? We've seen that ricin A chain without B chain is about 5 logs less toxic, which certainly says compartmentalization is something that's critical. However, maybe this will also point towards some cells being more amenable and a mechanism to why some cells are more amenable to transfection with just oligonucleotides. But I will leave that for the next. Thank you all very much.